Hello, my name is Mark Rami. I'm the team leader of Error 404 team. In this video, I'm gonna highlight the main points about our essay about FDA-approved coloring agents. First, we'll talk about what's meant by coloring agent. A coloring agent can be dye, pigment, or other ingredient that can give food, medication, cosmetic, and enable us to recognize medication dosage. For example, in hospital, where they identify different type of anesthetics by addition of different coloring agents. It can be known as also certified colors, which obey and controlled by FDA regulations. According to their source, which is our first classification, coloring agents can be natural, like mineral derived, for example, titanium dioxide, or plant derived, like saffron, or animal derived, like cochineal extract, which is derived from an insect, and synthetic coloring agent. According to their solubility, which is another classification, coloring agent can be a dye, which is soluble in water, for example safranin, or pigment, which is insoluble in water, like beta carotene, or lakes, which is also insoluble in water, like aluminium lakes.
In this slide, we're going to talk about the ideal properties of coloring agent that pharmaceutical companies take in consideration while manufacturing drugs containing coloring agents. It should be non-toxic and inactive pharmacologically, physically, chemically, and biologically stable, free from harmful impurities, should be odorless, unaffected by the light, temperature, hydrolysis, oxidizing or reducing agents, pH changes, and microorganisms. It should be also stable in storage and compatible with active pharmaceutical ingredients, and do not interfere with the test of preparations. Our first example of coloring agent is beta-carotene. As you can see, the conjugated system of its structure. It can produce colors varying from pale yellow to dark orange. It is generally soluble in chloroform, which is organic solvent, and insoluble in water, ethanol, and glycerine. It is incompatible with oxidizing agent, as decolorization will take place. It can be used as a color for sugar-coated tablets and suppositories. The second example is tartrazine. It is yellow or orange yellow powder. It is poorly compatible with citric acid solutions, ascorbic acid, lactose, 10% glucose solution, saturated aqueous sodium bicarbonate. It may cause allergic type reactions, for example, bronchial asthma, and also patients with hypersensitivity of aspirin. So manufacturers reformulated their drugs in order to be compatible with the new FDA requirements. The third example is titanium dioxide. It's widely used in pharmaceutical preparation as a white pigment. It has light scattering properties. For example, powder with average particle size to 130 nanometer scatter visible light, while average particle size with 16 nanometer scatter ultraviolet light and reflect the visible light. When mixed with methyl cellulose, titanium dioxide can reduce elongation and tensile strength of the film, but slightly increase the adhesion between pigmented film and the tablet surface. The first example of pharmaceutical formulation that use coloring agent is amoxicillin tablets, which use titanium dioxide as a coloring agent. There is other excipients like colloidal silicon dioxide, which is an anti-caking agent, adsorbent, and astringent. Magnesium stearate, which is a lubricant and release agent. Polyethylene glycol, which is a water-miscible co-solvent. Polyvinyl alcohol, which is a tablet coating agent. Povidone, which is a astringent and tablet binder. Pre-gelatinized starch, which are binders to improve tablet durability and integrity. Sodium starch glycolate, which promote rapid disintegration and dissolution of the tablet. Soy lecithin, which is a dispersing agent. The second example of pharmaceutical formulations that use coloring agent is Bolidigo, which is a sterile deep blue diagnostic dye for intravenous use. It contains indigotein disulfonate sodium, which is known as indigocarmine, and may also contain citric acid and sodium citrate to adjust pH value between 3 and 6.5. Here is a references page that we used to do the assignment. Also, I want to thank my colleagues, Fadi Samuel, Hamad Bey, Karim Rafi, for helping to do this assignment. And thank you for watching.